our BP Endure virtual seminar series. And today we have the privilege uh, of listening to Dr. Tracy Bale this afternoon about her research, especially the title of her talk is Identifying Novel Mechanisms of Trauma Programming. How do you cope with stress? And I just gonna talk a little bit about Dr. Bale. Dr. Bale is professor of pharmacology and director of the Center for Epigenetic Research in Child Health and Brain Development in the School of Medicine at the University of Maryland, Baltimore. She completed her PhD at the University of Washington in the Department of Pharmacology, followed by a postdoctoral work at the Salk Institute with Dr. Bale. Dr. Bale was professor of neuroscience at UPenn for 15 years prior to her move to UMB. So UMB got really good opportunity here. And her research focuses on understanding the role of stress dysregulation in neurodevelopmental and neuropsychiatric diseases and the sex differences that underlie disease vulnerability in humans and using the mouse as a model. She's interested in developing models of parental stress and the germ cell line involvement in the intergenerational programming of neurodevelopment. She serves on many internal and external advisory committees in National Institutes of Health, and she was reviewing editor of the Journal Neuroscience for almost 6 years. And she has been recipient of multiple awards in the area, including the Career Developmental Award for Early Career Achievement and Promise from the Society for Neuroscience, the Richard Weitzman Memorial Award for Exceptionally Promising Young Investigator Award by the Endocrine Society, and the Medtronic Award for the Society for Women's Health Research for outstanding research that has led to the improvement of women's health. And most recently, the other award is the Daniel H. Efron Award from ACMP. And recently, she was elected president of the International Brain Research Organization, IBRO. So thank you, Dr. Bale, for being with us today, and we look forward to your talk.
Thank you so much, Carmen. It's a great privilege, actually, to be here and to get to discuss the science, but also the piece of the science that we do related to, or the translation of, to human health. But also as part of the recruitment that you mentioned, Carmen, from Penn, which is in Philadelphia, down to Maryland, here in Baltimore, was part of this development of the center that focuses on both the epigenetic research that we do, but also child health and brain development and engagement with our community here in Baltimore. So I hope that that is something that the audience is interested in, and we can have a great conversation either during or at the end if anyone has any questions. So we are here to talk today about many aspects of neuroscience, especially our program in neuroscience and the great graduate group that we run here, as we're all interested in the brain, right? It's the most elusive and yet magnificent organ in the body. It is the most complex by far. And as a neuroscience researcher, I have found throughout my entire career that I remain every day fascinated by the research that is done and contributes to trying to understand the beginnings of the brain and all the way through across the lifespan of how the brain functions and its relevance, of course, to neuropsychiatric disease risk. Much of the research that happens in my lab is focused a lot across the lifespan, but especially the early period of brain development and neurodevelopmental disorders, but also how it just contributes to mental health risk. We are also very much interested in trying to understand those risk factors, especially for marginalized populations and those at risk for health disparities. So we know in the US that amongst African Americans and at the increased stress right now of our Black communities, that they are at a significantly increased risk for mental health disorders. The numbers from a few years ago are around 20% increase compared to white adults. And I actually was looking at some statistics related to my talk today in Puerto Rico with a lot of the stressors and traumatic experiences that have happened in recent years in Puerto Rico. And you can see from this recent data, actually this is, I think, from data from 2012 to 2014, so probably doesn't include some of the most recent disasters, but if you look at those reporting as adults, so 18 and up, prevalence of what's considered serious psychological distress, you can see that in Puerto Rico, amongst other ethnicities, is the highest rate and more than double of white non-Hispanics in the US. So it's an important aspect, and I know something that you're all very concerned about as well. And we take that to the next level of really understanding the biological underpinnings of how stress and trauma, and that includes discrimination stress, affects outcomes. I think one of the most awful numbers to look at is within the US, the maternal death rate. So we know that in the United States of America, that a woman of color who has even an advanced degree or education still remains to this day 4 times more likely to die in pregnancy or within the first year of birth, she and her infant, than a white woman with an 8th grade education. And why I make that clarification is because it's not limited to socioeconomic effects. Those, of course, and health disparities are incredibly important, but there is other factors that we need to consider here. And I think the impact of discrimination, stress, and access to resources is incredibly important. So on the bottom of the slide here, you can see the infant mortality rates. This is the state of Maryland, where I live. You can see that again, for Black babies, the mortality rate remains very high compared to all other ethnicities. And again, I pulled up some data related to breaking this down by ethnic backgrounds. Here you can see Puerto Rico, which I thought was interesting, that similar to non-Hispanic Black population, that Puerto Ricans also have a very high rate of risk for preterm birth and low birth weight. And those are major health considerations for not just survival, but also for the quality of life and other health risks. And we need to really begin to understand the impacts of these health disparities on the developing brain and their long-term effects on health disease and risk. So Arlene Geronimus, who is at the University of Michigan, coined a term called weathering. And I don't know if many of you have heard this term, but it's really important for thinking about the impacts of all kinds of stresses across the lifespan. So she coined it as the stressors that impact people of color are chronic and repeated through the whole life course. And this effect of weathering, that they accumulate these stressors and they have massive effects. This is just showing you an example of the impact they can have on mom who is pregnant, but also can be passed on to the next generations in terms of the biological influences on outcomes. So one of those key outcomes that we think about is called ACEs, or adverse childhood experiences. So Kaiser Permanente did a health study some years back where they investigated the question of, do these sorts of experiences across your life prior to adulthood, if they were to accumulate and we could identify what the major influences were, could we begin to identify everybody's risk for health and disease? And so what they determined was, and I'm going to point out here that these studies were initially done in a very white California population. Many individuals have since looked at these 10 adverse childhood experiences that were identified and have begun to add to them for other populations, especially urban environments that are non-white. But this study looked at, from the Kaiser Permanente, the 10 adverse childhood events and their accumulation. And what they found was the accumulation of the 4 or more of these ACEs. And it really didn't matter which 4, but 4 or more was the cutoff point that highly predicted health risk, especially for mental health risk, for things like addictions, depression, et cetera, and anxiety across the lifespan. Why does that matter? We live in the city of Baltimore. Baltimore is a majority Black city. And so when we start thinking about influences, especially during pregnancy, we want to understand the population that we have an obligation to help and to study. So if you look at pregnant women across the United States of America and look at ACEs that they've experienced, and I'm showing you here on the left, as 0 or all the way up to 4 or more, women who were currently pregnant when this study was done. And you can see in yellow where that break point for increased risk for mom and baby is located, that across the United States, it's about 12.5% of currently pregnant women. If you actually look in the city of Baltimore, that percentage goes up to over 61%. And what that says is that we need to really begin to understand those influences and those experiences unique to given populations that we're trying to study and determine risk factors And for the lab, really to begin to understand how can we model these outcomes and experiences, and how can we interpret in order to take that data back into the population. Okay, so I am a neuroscientist. I was trained as a neuroscientist, but it's become very clear over the 15, 18 plus years, whatever I'm at now for my career, that if we're really going to understand the developing brain, we need to understand more than just the current state of the brain. We need to really go backwards. We need to understand what's happening during gestation, a critically important time for brain development. And even those ACEs that I was showing you of mom. So mom's environment, as it determined even her intrauterine environment before she was even pregnant, which then takes us way back to trying to understand mom's influences that develop the egg. And as I'm going to show you today, also some data on dad's influences as it gives rise to dad's germ cells as well. That the accumulation of these events when they come together at fertilization kick off a host of biological signals that are really important for understanding not just how the brain develops, but everything from the rate of brain development, right? We know that at cellular division states, when it goes from the zygote, which is a fertilized egg, to a 1-cell embryo, to a 2-cell, 4-cell, 8-cell, all the way up to a blastocyst. And then we have all the determination of the factors after that give us a fetus. That by that point, you've already set in motion all of these factors, including really important epigenetic determinants that can guide things like the rate, how fast you develop. Do you develop too fast, which means you're missing some of the fine tuning that might happen in brain development, or you develop too slow, which might put you outside of windows of key influences and regulation of the brain. But you also might be experiencing then across subsequent parts of development, in gestation, postnatally, early childhood, adolescence, that all of these things become cumulative. So I like to say I am a neuroscientist, but really I'm a neuroendo, repro, placento, germ cell androgynologist, because we need to think about all of these factors when we talk about the brain. So I want to take you back today to, I hope many of you have taken in your cell biology or other classes as an undergrad, that we need to consider other factors along the course of development to really understand the points of risk. All right, so as Carmen pointed out, we have to, understanding these models, we cannot manipulate the human being, obviously. So one of the key models used in labs today is mice, and that's because we can manipulate the transgenics of mice to really get and understand the genes that are important for mammalian development. But we'd like to take this a step further in the lab, that we're not just creating models and studying the effects of stress in the environment on our mice, but we actually are pushing the limits of trying to understand and calling it a humanized mouse model. No, we're not crossing humans and mice, but we're trying to understand the factors that we can take from humans who we know are at great risk and applying them as best we can into mice, sorry. So the first story I want to tell you about is related to the microbiome. So you've all heard by now, I know, because it's in the lay press everywhere now these days, about your microbiome and how important it is in sort of your inner ecosystem. And there's many depots for your microbiome. We hear a lot about the one that's in your gut, but you also have a microbiome on your skin. You have a microbiome, which includes your armpit, right? There are actually investigators who specifically study the microbiome of the armpit, believe it or not. There's the microbiome of the oral cavity of your mouth. And there's also a really interesting microbiome of mom's vagina. So it turns out, ladies and gentlemen, that when you are born, for those of you who went through the vagina, the birth canal, and not C-section delivered, that when you are born, you pass through mom's vagina. Yes, that's true, you do. And in that process, you actually ingest mom's vaginal fluid it's sort of like a vacuum as you're passing through at birth. And as you're ingesting mom's vaginal microbiome environment, that microbiome is doing a lot of things that are really important. So one of the things that this work is being done in the lab currently by now was a postdoc, but now assistant professor, Eldon Jaserovich, and a team of staff in the lab. So he's begun studying the effects during gestation while you're developing, mom's gut microbiome, as you can imagine, is really important. The composition of that microbiome, everything that mom eats and everything that she's experiencing is assisting in the breakdown of that food and the microbes that are important for everything from how the placenta develops to all of the nutrients that the baby gets then. But at the process of birth, you actually get vertical transmission of mom's vaginal and fecal microbiota. Why does that matter? It matters because this is the first inoculant that the baby's gut is going to see, the first one. Otherwise, it's totally sterile. And what that does is it tells all of the immune system cells that are surrounding that gut that, hey, we're friendly here. We're going to educate each other and we're going to live in harmony throughout your life. Now, the composition of that gut for that baby is going to change substantially. It's going to change from the few days after birth, as soon as the baby starts nursing, because there's tons of lactobacillus, of course, in breast milk. It's going to substantially change as the baby develops and goes throughout life. There are many factors that will influence, but this initial stage is so critical for educating those cells that line the gut and especially educating the immune system so that you have a long-lasting effect. So you can imagine if there is something that affects the vertical transmission, you're born by C-section and you don't get this vertical transmission. So now your initial inoculant is going to be coming from lots of things in the environment. And that has been shown to increase risk for allergies. Okay, so anybody out there who was born by C-section, you might want to think about that. But influences of mom's life, such as trauma or stress, can also change the composition of her vaginal microbiota. And that is something that we're very interested in modeling. How do we do that? So Eldon started these studies almost a decade ago, maybe 7 years ago, really starting to figure this out. And we can C-section deliver mice. So we take a pregnant mom, and mice have a litter. So there's anywhere from 8 to 9 possibly in that litter. And we cesarean deliver them so they don't pass through the birth canal. And then what we can do is we can collect vaginal inoculants from control moms or moms that have been stressed during pregnancy. And we can then perform what's called an oral gavage here by using this little PICC line you can see. And I'm actually going to show you a little video that Eldon put together to show you how incredible this procedure is. These mice are teeny, teeny, tiny. And we can now take that PICC line and we can insert it in those pups and we can deliver what they would normally be getting by passing through the birth canal. So experimentally, this allows us to ask the question, if you get a control inoculant from a non-stressed mom, or if you get a stress inoculant from a stressed mom from her vagina, does it make a difference? And we can also vary the intrauterine environment. So we can take one of these little pups and we can take one from a stressed mom and one from a control mom, and we can switch. So you end up with 4 different groups. And Eldon did those studies and that work is all published. So I'm not going to go through it today, but he found that in fact, it is very important. It's important for the gut. It's important for the baby's immune system. And it's important for brain development because it shapes a lot of the information that the developing brain receives. So we wanted to take this a step further. We wanted to ask in an interurban environment where we know there's a lot of stresses, there's violence that's witnessed, there's shortages of healthy food, what are called food deserts availability. And the food desert piece of this is incredibly important. It's not just the absence of healthy food, but it's the absence of what's called a high fat, high salt, high sugar, but really importantly for the microbiome, it's a low soluble fiber. And if you are not up on your fiber intake, this is something you should look into. Your soluble fiber is so important to the microbiome that live in your gut. They need it. They need it to manufacture things like short chain fatty acids, which are critically important for the functioning of your brain. And the only way your body can get them is through digestion of soluble fibers. So you can imagine in an environment where mom is pregnant and she has a shortage of having access to those sorts of resources, what kind of a detriment that could be. So we took this model, and as I said, we wanted to develop something that was called humanized. So we were able, in a collaboration on campus here at the University of Maryland, working with a group in IGS, the Institute for Genome Sciences, to obtain human microbiome vaginal samples during pregnancy from women in Baltimore. Okay, so these are the different groups. I don't want to spend a bunch of time on them, but the take-home message here is that Eldon was able to develop a very complicated but fascinating model where he can vary mom's diet before she's even pregnant. So this is comparing high fat, low fiber, versus low fat, high fiber. So this being a very healthy diet, this being a very unhealthy diet. He was also then to track the animals before pregnancy. We wanted to make sure that they were heavy. The animals that were on this high fat diet were heavier, so there was a weight threshold. But importantly, that we did a glucose tolerance test because we also wanted to model the pre-diabetic state because that's also a risk factor for a lot of women in the urban environment. So we had these two groups, and this GV means Gardnerella vaginalis, which is a pathogenic strain of bacteria that promotes vaginitis. It is highly present in a subset of Black women, and it's associated with preterm birth. And we know that that's a big risk factor. So we're able to model all of this. So we do the pre-pregnancy setting with the diet. During the pregnancy, he can do a vaginal trituration, which I'm showing you over here, where he can take Gardnerella vaginalis from women and inoculate. So they're getting this intrauterine environment during gestation here of this litter is being exposed to mom's pre-diabetic obese state and that pre-immune environment there in her vagina. Okay, then we C-section deliver them, and then he can inoculate those animals with two different, what's called community state type 1 and 4. And why that is important is because community state type is a healthy vaginal environment, and community state type 4 is one that is associated with preterm birth. And these are obtained from human beings. So humans, women who are pregnant, the samples are obtained and we're inoculating mice, right? Because a microbiome is a microbiome. It doesn't matter who the host is. So what is fascinating from all of the work that I don't want to spend a bunch of time on today is the first thing that we found with this, what we're calling triple hit. Triple hit, so pre-diabetic, pre-obese state, Gardnerella vaginalis, and then the inoculant. You'll see here is we created a 60%, almost 60% infant mortality rate, which is truly amazing. And it happens within 48 hours. If you survive 48 hours, then you survive. So it's really the immune system that's taking a big hit here. And you can see that the difference is the biggest impact is that low fiber diet. And then if you sort of back off one at a time of those 3 hits, you can get back up to 100% survival, right? So the biggest hit here is the diet, that low fiber piece. And if you survive, what's really interesting is you can also see this increase in body weight that post-puberty, those animals that survive this triple hit, they take off post-puberty with increased body weight. And that is a risk factor for diabetes and obesity that we're looking at now. And I just wanted to show you, just for quick illustration points here, the difference in these two diets. This is a measurement looking from CyTOF mass cytometry, which is a way to look at all the immune cells in this population that Eldon was working on. And you can see the difference just by the level of, just pay attention to the bright orange or this reddish color here. On the top is the low-fat, high-fiber diet. This is looking at the pups just as they're born, as we're inoculating them by C-section. And you can see on the top, huge differences in the immune cell population. So basically, this is what a maturing young immune system should look like. Look at what happens when you give a high-fat, low-fiber diet to mom. These animals are born with a raging, mature, and very angry immune system. And that real increase in neutrophils, which is shown over here, can be very dangerous and is also highly associated with infant mortality. So I'm going to end on that topic and all the work that Eldon's been doing to jump to our next topic, which is, okay, that was mom. But what about dad in the interurban environment? What about all the stress and trauma that dad has experienced across his lifespan? What do we know about that? And that's a much more complicated question. So we know historically that dads and their lifetime experiences with stress have come a lot from the Dutch hunger winter. We know towards the end of World War II that there were individuals, especially within Holland, that were experiencing very low caloric availability. So they tracked a lot of the outcomes from these children, their offspring, and their grand offspring to look at the intergenerational contribution. And what they found is, in fact, that there was evidence that boys who experienced this famine during given periods of development passed on differences in health risk, especially related to mental health, to their offspring. And so also there's the Swedish famine studies that you've probably heard of. Up here in the north of Sweden, this Överkalix region, they're so isolated that for centuries, they've kept all kinds of amazing church records for all of these different areas. And so the church records have been mined for things like famine years, feast years, right? Because they couldn't go to the grocery store if they were running out of food. And so they just starved. And so they also kept all kinds of health records and birth records so they could look at all of this data to begin to build associations. And so similar to the Dutch hunger winter, they also found from this data in this Overkalix region that there were periods for boys who experienced famine versus years of feast, that they themselves had differences in health and disease, but they also passed on similar effects. Some of the most amazing data has demonstrated that famine exposure during periods of development for those boys passed on effects for longevity, meaning that you tended to live longer, up to 30 plus years longer on average, depending on your dad's experience. Right? So those are association studies, but they give and provide evidence that this is really important and something for us to consider for not just mom's influence on the fetal brain, but dad's influence as well. So this has become something also the press is very interested in. Highlighted our work from the University of Maryland here and the program in neuroscience. Graduate students who are doing this work in the New York Times recently, I guess we're looking at almost 2 years ago, where they started to ask this question about men. So not just women and pregnancy experience or adversity during preconception environment, but dad as well. And this actually was a study that was looking at some outcomes from the Civil War and how you pass on intergenerational effects. So why is this important? Okay, so we're neuroscientists, but here we are back at the germ cell. So dad's germ cells are generated in a continuous way, which is very different from mom's germ cells. And we know that the somatic cells, those cells that are not germ cells that line the reproductive tract, are really important for the development of maturing sperm. But if you look within a sperm, a mature sperm, and you were going to say, okay, so what epigenetic marks, these modifications of the DNA, whether they be on potential histones, which there are actually very few of, there's more protamines, if you remember from your cell biology class. The histones are swapped out for protamines, but there are modifications. There are DNA methylation marks, although post-conception, most of them are erased. So the question really became in this field over the last decade, okay, so dad's experiencing something in his environment. He's been traumatized, he's been stressed, he's had an infection, he's experienced a famine. Sperm are transcriptionally inert, mature sperm. They cannot respond. How is this happening? And if we could get at that biology, can we then ask the question in humans to identify risk? So my brilliant graduate student, Jennifer Chan, developed this model. This is just showing you, again, here's a testes. This is true for all mammals. On top of here is the epididymis. And so when sperm leave the testes at the end of spermatogenesis, they're not mature, they cannot swim, and they cannot fertilize yet. They have to they actually enter into the top of what's called the caput, the head of the epididymis here. And despite what it looks like here, it's actually comprised of these tubules. So inside the tubule, swimming through the little channels, is sperm. And they enter this process, and they're there for quite an extended period of time, weeks, where these epididymal epithelial cells that surround these tubules, these orange cells here, they're called epithelial cells. They're secreting all kinds of growth factors and nutrients, and they are essential for the maturation of the sperm. Without them, the sperm will never swim and they cannot fertilize. But in addition to those growth factors that are being secreted from these cells are what's called extracellular vesicles, these teeny tiny little vesicles. So what is an extracellular vesicle? An extracellular vesicle is a teeny tiny membrane raft or lipid bilayer containing particle. It is not a cell, so it has no ability to promote transcription. But what it can do is it can contain all kinds of cargo that are being released. So all of these vesicles, you may have heard of exosomes. An exosome is a specific type of extracellular vesicle. It's one of the smaller ones, so it's delineated by size. But they are released from a cell from all different types of intracellular compartments. They comprised of really interesting different proteins in this lipid membrane, but the intraparticle compartment contains a lot of small non-coding RNA, such as microRNA. Okay, extracellular vesicles are fascinating because they are released in all mammals from all tissues, including the brain. They travel at high concentration in circulation. Their proteins are associated with all kinds of things, but they especially are associated with signaling the immune system. And at the end of the day, we all love the brain, but really everything seems to factor and whittle down to the importance of integration with the immune system and keeping us healthy. So how do all of the tissues in your body signal the immune system and communication? A lot of it is extracellular vesicles. And in fact, going back to Eldon's microbiome studies, there are specific types of extracellular vesicles actually released from bacteria in your gut. Okay, so all of these fascinating extracellular vesicles are in circulation. They travel to all your tissues. When they arrive at that tissue of specific action, so they have really specified proteins in the vesicles from where they're released. It's sort of like a letter, right? The to and the from. So they act at specific sites and they perform specific function. So Jennifer Chan developed this model to ask, okay, we can't isolate these extracellular vesicles that are being secreted here because a mouse's epididymis, the tubules, are just too teeny tiny. You could never get a specified, right? We don't want to get the circulation. We want to get the specific ones that are interacting with the sperm. So this brilliant graduate student spent many years developing what we call stress in a dish. She was able to culture mouse epididymal epithelial cells from the caput, that head of the epididymis. And in doing so, she can ask very specific stress questions where these cells then release into the conditioned media here, extracellular vesicles that we can capture. We can take those vesicles, and what I'm showing you here is Jen's beautiful work, which is all now published, looking at the protein content by what's called proteomics. So this heat map is turned on its side just because of space issues. But on the top are extracellular vesicle proteins from control vehicle-treated cells. On the bottom are cells that we use the stress hormone corticosterone to treat. I don't want to go into all of the different parameters, but I think you can clearly see that, first of all, each column here on its side is a different treatment, different cell culture dish. Each row then is a different protein. And by proteomics, you can see that those proteins that are lowly expressed in vesicles in this group are very highly expressed in this group. And those that are more highly expressed here are also lowly expressed here. So there are thousands of proteins, some of which, as you can see in the top here by hierarchical clustering, are almost absent in one group and very abundant in another. And that's important because it's telling you something about the signaling process of these vesicles. Okay, so I'm going to jump to the end of Jen's thesis that she did, where she did this amazing experiment. She collected these vesicles, what I'm illustrating here. Those were from the prior vehicle treatment, and those that were from the prior quart treatment. Now, they've obviously been through 8 days of washing. The quart is long gone. The cells have been changed. Media's been changed 3 times. It's 8 days later, okay? So she's now collected these extracellular vesicles. Now, she wants to know, do these vesicles do anything, right? That's the ultimate question. Do they do anything? Okay, so this brilliant graduate student then took a normal control mouse dad, right? She collected his mature sperm from the caput region that was maturing there. She divided it into 2 pools. So we know that the sperm coming from this dad are identical. She incubated half of his sperm with the vehicle or the control vesicles and the other half with the red ones, which are the previously quart-treated vesicles. She then washed the sperm. She then took a normal, healthy female mouse and she collected eggs. So all of the eggs coming from the same mom. All the sperm from the same dad, all the eggs from the same mom. We then performed intracytoplasmic sperm injection, which is a normal fertilization process, even used in humans. And you can take an egg and you can take a sperm and you inject it into that egg. And now you can ask the question, does the extracellular vesicle content matter for what happens in the development of those offspring? And the way that she asked that was, she then transferred these fertilized eggs, these zygotes. You culture them for a period of time and then you can transfer them. So she did multiple experiments, one of which is she transferred them either to the left side or the right side of the same mom donor. Okay, same sperm from the dad, 2 pools, same eggs from the same mom, injected. Now you're either developing on the right or left side, meaning everything is identical in this environment with the exception of the vesicles you were incubated with. And then she also followed up with subsequent experiments to ask about the outcomes. I don't know what happened to this data. Sorry, I lost the data. But just want to tell you that this is amazing because what she found was that midway through gestation, if she takes out the placenta or the brain, either one, there were dramatic differences in neurotransmission genes, meaning the brains were developing likely at different rates. And we know that stress in the environment can dramatically shift the rate of development. And that matters Because if you're born too fast, just like if you're born too slow, you miss out on a lot of precision and tuning of the brain. And so the data that I'm also not showing you, because I deleted it accidentally, but this is showing you in the offspring that both in males and females, it didn't matter the sex of the offspring, that these animals showed differences and actually replicate Jen's findings if dad had been stressed and bred normally, that their stress response, the offspring stress response, is changed. So all of this is to say, to demonstrate the causality of just one stress signal, these extracellular vesicles, and how it might shape development. Okay, how do we know anything about the clinical relevance of this? There are many things that you can question here in the mouse. It's still a mammal, but it's not a human. Humans' lives are much more complicated. But we set out on a study. Chris Morgan actually has been working on these studies for several years now. And again, in a collaboration with Neil Epperson, we began when we were at Penn and have now continued, and these are also recently published. We actually took Penn undergrads, a relatively homogeneous environment, and we wanted to ask the question, if we were to track perceived stress and take sperm donations each month, because we don't really understand a lot about the composition of human sperm. So if we're going to look at the contents, the microRNA content of sperm as would have been delivered by those EVs, how do we get at that question? So we did all kinds of characterizations through the Department of Psychiatry with Neil Epperson at Penn. We followed their perceived stress each month. So for instance, over final exams, a big stressor. We then did small non-coding RNA sequencing. So not just microRNA of the sperm, but also microRNA, piRNA, and tRNA fragments. A huge, very complex bioinformatic assessment that then Chris modeled looking at within subject. So how normal every month is your small non-coding RNA composition of your sperm? And how does that variability correspond across subjects, right? That's two big questions. So this is a snippet of some of his beautiful data, which turned out to be incredibly interesting. And believe it or not, with only a subject pool of 20 men giving 6 monthly donations, he found significant outcomes from microRNA, piRNA, tRNA and piRNA is here on the bottom. This is just showing you an example. Each number here is a different subject. So this was person number 13, person number 2, 22, etc. What you're seeing here in the dark blue across the top here in A is microRNA populations. So Chris was able to model based on within and between subject, that dynamic composition. What you can't see in the background maybe is the light blue. So of course there are hundreds of these microRNA in human sperm. And what is showing you in the dark blue are those that rose out as being highly expressed and variable. And so you can see in the microRNA, you start to see some patterns across subject. We identified some that were common across all subjects. The tRNA fragments, of which there are far fewer. But look at this piRNA, never discovered before, because piRNAs, which are expressed highly during spermatogenesis, evolutionally very important for the process of protecting dad's DNA, never known before to be present post-maturation. And look at this composition and look at this pattern, which is telling you over the course across all subjects, right? It's 6 months for everybody, but it could be a different 6 months depending on when they started in the study, suggesting possibly a whole different change to dogma about what we understand of composition of sperm and what they might be doing related to, who knows, fertility. We don't know what it means, but you can definitely see that pattern there. So fascinating. So I'm going to end there and happy to take questions, but I want to just drive back this really important idea. So I'm going to end with the data there, that the child's brain development, very important across a host of biological signals, everywhere from mom and dad's germ cells, across early development. I didn't have time today to talk about the placenta, but all of those factors that influence mental health outcomes and infant survivability. So just quickly, because we're running out of time, I promised you that I would say a little bit about our center. So all of this, sorry, are we okay? Hello?
Yes, we're okay.
Okay, so I just want to quickly mention the center. So all of these translational studies that we do, as I've tried to highlight some of the work in both in our rodent studies, in our cell culture studies, but also we do in humans. So all of our mouse models also have a human component to them, is because we're very interested in engaging in the Baltimore community especially, but we have collaborations, frankly, all around the world. where we look at different outcomes related to stress and trauma. So in the Baltimore community, as we are a majority Black city, this is actually a community, a marginalized population with lots of health disparities. And we know the history of the Baltimore community as it has been impacted by many of these outcomes important for stress and trauma. So we've enlisted multiple ways of engaging with the community. I'm showing you on top here a picture of a bunch of our kids that we work with in several of our elementary schools. This is actually the mayor of Baltimore who came to work with us one day in our science and art project. So we do a lot of community enrichment through the schools to engage with the kids, the school itself, their families, and the surrounding community to talk about science. We use art and science to communicate the importance of the brain, and we use reading as one of our positive outcomes for its incredible benefits to the young brain. We also do science and society events. This is showing you Greg Dunn, who's a neuroscience artist, and some of the work that's actually hanging in our building that we tasked him with providing for us that speaks to the real big importance of early events as it gives rise to our brain and interactions with the environment. And this is actually one of our city council members, Zeke Cohen. So I've given talks at Baltimore City Council, and we've talked a lot about the brain and trauma and its effects on the brain. Zeke actually helped pass the Baltimore City Trauma Act, which informs all things from education to health to to incarceration, et cetera, and the effects of trauma and the impact it has and how its consideration is vitally important for this community. So I'm going to end there. I'm just going to show you a quick picture of our mural. This is how we ended our year with the kids at this elementary school. You can see again some of our leadership kids in the front row. This is the principal of the school. This is our dean of the School of Medicine, some of our investors in this program. This is me. This is our famous artist, J. Wolf Schlossberg Cohen, who helped us and the kids with this mural. And this is actually the CEO of Baltimore Schools that participated as well. This is what our school looked like when we started at this elementary school. And after a year, this is how we left the school, though we never really leave the schools, of course. But then again, so that the community can see the importance of engaging, the importance of the brain. You can see all the neurons that are highlighted in all of these kids on the wall of the school, work that we're incredibly proud of, and the kids incredibly proud of the work that they contributed to that. And I just want to end with this piece for the center. This is actually our floor on the 9th floor of our building, where we brought the same art from the mural into our hallway so that we can engage with the community and remind everybody as the work that we're doing in our research, why it is that we do what we do every day and how important it is to the community. So just as a reminder and the beauty of the art that these kids contributed to. So I want to really take 10 seconds here to highlight the important work I showed you of Eldon Sharovich, and he has worked with a number of people in the lab and all of those microbiome studies because it does take a village. Those C-section models that I showed you, as you saw from that video, are really complicated. So Trayvon Giles is actually a student who's participated, who's working in the lab, who's actually off to graduate school with Eric Nestler shortly. Liz Hill has actually gone off to graduate school as well, was working on these. Lily Folt is a tech in the lab. And I also want to highlight some of the work done by Jennifer Chan, who's now a postdoc, also happened to be at Mount Sinai with Ian Mays, and some of the other members of the lab that have contributed. Chris Morgan, where's Morgan? Chris Morgan is actually doing all of the human studies. And again, we have so many great collaborators that have helped us with all of these outcomes. Please follow us on Twitter with all of our community engagement work, as well as the research that we follow. And of course, none of this could be done if NIH wasn't invested in funding this research. So I'm happy to take questions and answer anything that you're interested in about our program.
Wonderful. Thank you, Dr. Bale, for this amazing talk and also for showing how science can become part of the community. And I think that's so important and something that we are aiming at training our students to become, engaging in the needs of their social group, obviously.
Sure, absolutely.
It's, we, at least I think that educating our students to become citizens as well as scientists seem to be the right combination for developing a commitment to change.
So I could not agree more, Carmen. And I think it's essential to think about Sometimes we get in the lab and our exciting research, we put blinders on and forget the individuals, the taxpayers who are paying for that research, and our obligation to engage with them, not to just discuss the science, but that they are the ones benefiting from the science.
Exactly. So we have a couple questions actually, and the first one is from one of our students, and she says, if a baby is born vaginally from a surrogate mom who is not the biological mom, can that create contrast in his or her neurodevelopmental, considering his or her genes are not related to the vaginal fluid?
That's a fascinating question. Certainly not something that we have studied. The one thing before I finish answering that I want to highlight, because this is really important, especially with many of the mouse models that people work on, including us, that what we're showing you for outcomes of the brain is changed, very important. But really what we're saying is, if you think about, if you're going down a slide, remember from your childhood going down a slide, we call that sort of like the canalization, right? Your genetics determine the majority of how the slide that you're going down. But the walls of the slide, how you might extend up without going over, et cetera, that are guiding you and limit sort of the outcomes, those can be influenced by your environment. So while being born from a surrogate isn't going to change your genetics and that health risk, right? If you have a risk for addiction in your family, you have a risk for cardiovascular disease or breast cancer, your environment can change how you slide up or down going down that slide, but it's not going to change the slide that you're on, right? So we all know from our families what many of our risk factors are. And if we live healthier lives, it decreases our risk, right? But we're only talking about a risk. So the data I showed you, with the exception, of course, of infant mortality, that's beyond a risk, right? That's a very different outcome. But in terms of the brain and how it develops, I think it's important to keep that in mind, that your genetics are determining a lot about, those are your small nucleotide polymorphisms, your SNPs, that you can do on 23andMe or any of those outcomes, right? So that isn't going to change no matter who gives birth to you. Well, the DNA of mom's and dad's germ cell, ultimately. So I will answer it that way, but very good thinking. It's a fascinating question.
Good, I'm glad. There's another question that says, if a mother with depression or with stress can transmit this to the baby?
So that's the question. So can mom with depression or stress transmit to the baby? So mom's depression or her neuropsychiatric or neuroaffective state risk, which is, again, the former director of the National Institute of Mental Health, Tom Insel, frequented the term that I like, which is, a lot of what we experience in life is a G by E by D. Your genetic background, which we cannot change, by your environmental experiences, by those developmental windows. And what that means is, again, you're going down your genetic slide, and those environmental experiences. So if mom's depressed during pregnancy, and all of the factors related to changes in her immune system, stress hormones, things she's eating, changes to her microbiome, those are all factors of that E that can shape your development. And that D, the developmental windows. So if she's depressed during pregnancy versus, let's say she's depressed during your early developmental years postnatally, right? Those are the developmental windows we're talking about. So they all shape us. Absolutely, they shape us, but they're not dogmatic and they're not predetermining because mom had depression, you'll have depression. They're contributing risk factors. And I also want to highlight, they're also contributing resilience factors, right? We also know that a lot of individuals, whether it be genetically or environmental information, can also make us resilient.
Wonderful. Then I have another question. I was wondering if you have looked at the effects of inoculation with the different vaginal microbiomes on animal behavior later in their lives. For example, anxiety-like behaviors during adolescence.
Great question. So we have looked at in Eldon's paper, which is Nature Neuroscience, a few years ago. I'm trying to remember the year. Doesn't ring up. 2017, 2018. His Nature Neuroscience paper has all of that initial data where he looked at the effect of those inoculants to shape those sort of anxiety-like or related outcomes. It very much shaped, if mom had been stressed, especially during early pregnancy, changes in the composition of her vaginal microbiome contributed to changes in the offspring brain and development that made them more stress reactive themselves. Again, just a risk factor, not something that's going to necessarily determine disease. But if you're different, if you respond to stress in your environment differently, you can now imagine the augmenting, the accumulation of that effect. If you're now more stress sensitive and you experience stress and so on and so on. And he also ran those animals through all kinds of outcomes. So it does shape their risk factors for things like cognitive performance and anxiety-like tasks. Yes. He has not yet done that with the humanized studies. These are currently happening in the lab right now. So he hasn't yet run those through any of the battery of tests. He's really been focusing a lot, especially with that infant mortality outcome, what the immune system effects are.
Okay.
Great questions.
Yes. And the other one is that what would happen to a baby that the mother had depression and the father sperm show, well, depression? I think that
Sure.
Both are depressed.
So these actually are studies that we've sort of touched upon a little bit. And some of the transcriptomic work is also recently published. This is actually Yasmin Cisse published this work recently in Placenta, just looking transcriptomically. So we actually did studies which were fascinating. We stressed preconception. So we stressed a female mouse, we stressed a male mouse, many mice, and then we bred them in all combinations. So we had control, stress, control, and control stress, all combinations. And then we studied outcomes related to behavioral outcomes and stress outcomes and transcriptomics, mid-gestation, all those kinds of things. Behaviorally, the differences are not quite as robust as they are just in terms of stress reactivity. There are differences. But what's super fascinating is if you look transcriptionally, by mid-gestation, whether mom has been stressed or whether dad has been stressed matters a ton for, again, the rate of the development. And if it's mom has been stressed, there's all kinds of differences related to metabolic outcomes if you look in the developing fetus. But if dad was stressed, all of those outcomes are related to immune system and a huge inflammation. So very, very big differences.
Wow, that's so interesting.
Yeah, it is interesting. What it means, still to be determined, but interesting.
Okay. Any other questions?
I have a question regarding the sperm transfer. Where the sperm, when they were transferred, the exosomes were not transferred. They were eliminated before transfer.
Good question. Good question, Jose. So what's super fascinating about this process, and so I've given talks, around the world at various conferences. I get invited a lot to reproductive conferences, and I was sharing a shuttle ride to the conference one day at a reproductive conference with a fertility expert, right, who works in the field of reproduction and fertility in humans. And he was asking about our work, and he became all of a sudden very concerned. And he said, "Oh my God. So in human studies with fertility issues, whether it's IVF or ICSI, whatever the procedure is, the first thing that they do is they wash the sperm." And he was panicking because he said, "Oh my God, what if we're washing away all of the vesicles that are important for signaling?" I'm like, well, so this is something that we're currently doing. I don't have an answer for you. Some people believe that the vesicles are taken up by the sperm. I can't tell you because these studies are really complicated to sort of be able to visualize. The tools don't exist yet, although we're working on some tools now. Some really cool 3D printing that we're working on to be able to visualize this process. I don't see how they could be taken up because the sperm head would just be enormous by the time it reached the egg if that was the case. I think it's more of a sticking process. That vesicles, it's like a homeostatic kind of process. If you think about, common pharmacology, and as they're transiting dad's reproductive tract and then mom's reproductive tract at the point of fertilization, that it's a process of environmental signal accumulation. And that when all of the sperm that reach the egg, and remember, one sperm fertilized, but millions of sperm from dad can reach the egg. And so all of those signals that they might be contributing. But it's a great question. We don't know.
Thank you.
Okay. So I think we're done with the questions for now.
Do you want me to stop sharing here?
Yes, please.
Okay.
So now we're being joined by both Brian and Georgia from University of Maryland that will be also now starting the part of outreach, not outreach, but network about the graduate programs at the institution.
Thank you, Carmen, and thank you, Tracy. That was a great talk. Now I have the specific challenge of following that amazing talk and even more amazing questions that you all had. That was really quite invigorating. Can you guys see this?
Yes, we can.
Okay, great. So I'm Brian Mather. I'm an associate professor in the same department as Tracy, Department of Pharmacology at the University of Maryland School of Medicine. And I'm also the director of graduate education for our program in neuroscience. So we are affiliated with the University of Maryland School of Medicine, same campus. And we're under this sort of umbrella graduate program in life sciences entity, where there are several other programs in molecular medicine, biochemistry, epidemiology, et cetera. But we are the program in neuroscience, so we will be talking about that today. Because we are affiliated with the School of Medicine, we offer PhD and MD PhD degrees. So I will be giving you a brief rundown of the nuts and bolts of our program, and then I'm going to pass the baton, so to speak, along to some former directors of graduate education of the program, who will tell you about different aspects of what's going on here, exciting aspects of what's going on in the program, and then we'll finish up with Georgia, our program coordinator, who's going to give you some information on student life, and then we're actually going to have some students give you just a little bit of flavor of what it's like to be a student here in the program. So where are we? As Carmen and Tracy were mentioning, we are in Baltimore. Baltimore is a major city on the East Coast near Washington, D.C. And I'm bringing this up because University of Maryland is a big place, and it's across multiple campuses. So when you think of University of Maryland, a lot of people think of the undergraduate campus, University of Maryland College Park, which is located right outside of Washington, D.C., where this red X is. And that's where they have all the big basketball and football games, et cetera. But we're here in downtown Baltimore, where this blue X is, with the rest of the professional schools in the University of Maryland system. So we're in the medical school, but we're on the same campus as the dental school, the law school, nursing school, school of pharmacy, et cetera. So like I said, we're in downtown. We're just a few blocks away from the Inner Harbor here, which is the heart of downtown. It's a beautiful place to go and think about science, to do science. Specifically, our campus is here, these buildings.
So Brian, if you go back a slide, that's actually a view from my house. I'm just going to point out. There you go. You can actually live in Baltimore with that view, and I'm not kidding.
Right. Tracy lives the life of a rock star, indeed. So in the foreground is our campus, and just a few blocks away is the Inner Harbor, and Tracy's house is right about here. So in campus, we have, like any other school, really, lots of amazing buildings, some really pristine lab space for you all to come and perform world-class research. So who are we? We have about 61 total students in the program currently, 7 MD PhD students, and roughly about 115 faculty at this point. Our leadership here is on the bottom. Margaret McCarthy is our director of the program in neuroscience, which cuts across more than just the graduate program. She is also my chair and Tracy's chair of pharmacology. This is me here in the middle, and then Georgia Rogers, who will be speaking a little bit later, as our program manager. So this is basically what it looks like to get a PhD here in our program. It's not very much different than a lot of other programs. So it's broken down here by year going across in these blue boxes, and then the top is the fall and the bottom is the spring semester. In year one, you take a core course, which gets everybody sort of on an even playing ground with the curriculum. And then immediately into the spring semester, you start your first of 3 laboratory rotations while taking a new course that we have developed called Advanced Neuroscience Investigation that concentrates on applied learning and developing experimental design skills. In year 2, in the fall semester, you take what's going to be a combined biostats and pro seminar course, which instructs on how to develop a hypothesis, write a specific aims page for a grant, and to tackle data sets using statistical methods. And by that time, you've chosen a laboratory for your thesis research. So at the same time, you're already performing thesis research. And then in the spring semester, early in the spring semester, you complete your qualifying exam and then continue on with research throughout the rest of your time in graduate school. And we have a roughly 5.5-year graduation rate. So admissions, you probably have already heard this already, but we do not require the GRE. Our deadline is December 1st. And as far as reviewing applicants, we take a holistic approach to admissions. We focus on, probably mostly focus on applicant passion for science, research experience, and letters of recommendation. We do require a personal statement, and that is where you can sort of convey that passion for science that you have. So with that, I'm going to pass it along to Jessica Mong. She is professor of pharmacology in the same department. She is the former director of graduate education for the program in neuroscience, and she's going to be telling you about financial support for our students. So Jessica, please go ahead.
Thank you, Brian, and good evening, everyone. So I don't have any slides to share, and I won't take much of your time, but I just wanted to let you know, if you aren't already aware, that in our program of neuroscience, we financially support our students throughout the tenure of their degree. I think most graduate programs do this, but just in case you were unaware of this. And as Brian mentioned, I am the current PI of a T32 training grant that is partnered with other indoor programs, specifically the newest one at Temple. And what the advantage of having a T32 training grant allows us to do is it allows us to add more training slots or positions for students. So we currently can bring in 6 students on our training grant. And there are other advantages to this as well, in that you get 2 full years of support from the training grant, plus if you're appointed, as well as travel funds. But I think it's important to make a really clear distinction that the training program, while it will support 6 students, the training is across the board equal for all students in our program. It's just that our program, I think, is very fortunate to be involved with the NIH training program, in that it adds extra support and allows us to give more professional development opportunities, as well as other outlets for our students to learn and grow as neuroscientists. So there are 2 other opportunities that I want to specifically mention. One is for graduate fellows, and this is called the Meyerhoff Fellowship Program. And I don't know if you were attending the Indoor event maybe 2 weeks ago, you might have heard a little bit about the Meyerhoff program from the keynote speaker. But what this program is it's 1 year of financial support, sort of a scholarship, but it is specifically designed and focused to enhance diversity in our graduate programs. And the neuroscience program has been fortunate to have a number of Meyerhoff scholars, two of which are going to be speaking with you in a few moments. But in addition to the financial support, and I think that this is the real advantage of the Meyerhoff program, whether you are financially supported by it or not, as a member of the Meyerhoff program, you actually do get interaction with diverse students across the campus, as well as monthly scientific meetings that allow you to explore, again, your science with your peers. And again, I think that Lace and Soad will be talking more about that specifically. And then the last program that I wanted to mention that is associated with the University of Maryland is called the STAR Prep program. And what the STAR Prep is it's a post-bac program that, again, is specifically designed for underrepresented student groups to sort of get them into the pipeline of science, and not only science, but neuroscience, as well as the University of Maryland. And so this is a very specific program that you apply to work in labs at the University of Maryland, I guess after you are finished your undergraduate degree. And the advantage here is that, like I said, it is the neuroscience program does participate in this. And in fact, it really has allowed students in the STAR Prep to get some research experience, sort of get some real foundations of what it's like to be in a lab full time. And we have actually accepted a number of those students into our program for a PhD degree, and they're actually doing quite well. So I'm going to end with that. I'm happy to answer questions when our presentations are over. And I will turn it over now to Dr. Mary Kay Lobo, who is also a former director of the graduate program. Mary Kay.
Mary Kay, you're on mute.
Sorry about that. Okay, hi everyone. So I am, we recently formed a diversity committee for the program in neuroscience, and I am now the chair of that new committee. Really, this committee has multi-purposes. Our main goals are for training in the students in the program. One is to, although we do have a diverse group of students, we know we need to do better. So we want to improve our recruitment of diverse students to the program. We also want to, we realize that we need to start educating our students about bias in neuroscience, in research, and in our community. So we started doing that, including we've done this now in a 1-credit course I direct this fall. We actually just had our session on racism and bias and promoting diversity in neuroscience today. Apologies for my dog in the back that's getting upset with me. And then we also want to improve outreach on neuroscience and diversity to our community, and just achieve equality overall in our program. So one great thing about our diversity committee is that it's composed not just of faculty. We have a number of students on there from diverse backgrounds, and we also have staff on there from diverse backgrounds. So we think it's important to listen to everyone's voice. And we have a number of things going on. One thing we just started doing, we sent out yesterday, is we have a PIN diversity memo that we send out with 5 pointers on how to educate yourself about bias and racism in neuroscience and promote diversity and inclusion. And we highlight some of our students, including the 2 that you'll meet today. So I'm happy to answer questions later, but I will turn it over to the program manager, Georgia Rogers.
Hi, everybody. Thank you for coming tonight and learning about University of Maryland. I was once in your shoes where I was looking for a graduate school. I have the unique position of not only working here now, but I actually am a graduate of this program. And I remember thinking, oh, Maryland, I don't know if I even want to look at them. And then when I did, I was wowed, and they sucked me back in. So I haven't left yet, it seems. I want to tell you a little bit about our students. First, I just like to identify myself as a person that's here for student needs, no matter what it is, from before you even apply to as you are coming up with your career goals as you graduate. So anything you need, whether it's about the application process, about our Star Prep program, about our actual PIN program, please email me. Marimar put my email in the chat. Thank you very much, Marimar. So I'm very responsive and I'm happy to answer any questions. So our students, I can't say enough about how great they are. Our entire community actually is quite close-knit, whether it's students, postdocs, faculty, everybody knows everybody by name, by first name, and lots of nitty-gritty details about them too. We have a lot of different events that bring us together. Some are obviously on a more scientific basis. We have plenty of seminars, we have retreats, things like that, but we also have social events. Case in point, tomorrow? No, in 2 days, I need to figure out my Halloween costume for our Halloween trivia that is coming up. So lots of fun things that keep us together. Our students actually benefit from not only being in our program, but by being under the umbrella program, the graduate program in life sciences. So they take one of their first main courses alongside of other students that are studying biochemistry, molecular biology. This allows for them to already start making connections with different colleagues, and really it's the science that benefits from those sorts of connections that are made. We really respect the voice of our students. We have a student training committee where we regularly hear feedback based on our curriculum, based on whatever is going on in the program. How can we make improvements? How can we make it better? And we don't just survey them and throw that information out the door. We actually make changes based on what we receive. We also have a neuroscience outreach volunteer association. So like Tracy was talking about some of the outreach that she does as part of her center, we have an entire association that does neuroscience outreach in our community, and that can look like many different things, from going into the school systems and teaching students about the brain to actually having bingo nights at a psychiatric hospital, because that's a very underserved population there, and just doing outreach like that. So to tell us more about student life and about students, what it's like to be a student in PIN, we have Lace. And so Lace, would you like to start?
Thanks, Georgia. I'll tell you guys a little bit about my background, both in research and kind of what led me down that path, and then some of my kind of unique takes on the program and why I think it's really strong and worth considering. So I'm just starting my 5th year in the program in neuroscience here at the University of Maryland. My dissertation research is focused primarily on understanding how we can better treat psychiatric conditions like depression by using antidepressants that are specific, rapid, and hopefully lack side effects. So I'm using compounds that appear to have those properties, and then I'm investigating how they work so we can design even better treatments. This work is a direct extension from the research I did as an undergrad at California State University in San Bernardino, California. There I studied risk factors associated with adolescent onset depression and the long-lasting effects of antidepressant treatment when given during that period in development. So why did I become interested in this particular area of research? Well, I actually didn't know that I would end up in science, at least not until the final year of my undergraduate degree, when I did become involved in preclinical research. But what got me to that point was wanting to see improvements in the way that stress and trauma-related conditions are treated. Because what I found to be particularly unfortunate, both in my family and in my community, is that some people resort to unhealthy ways of coping when they do not respond or have access to treatment. And so this is something that I've been very passionate about for a long time, because I grew up in Section 8 housing, where illicit drug use was a routine and accessible way for people to deal with the struggle and hardship of everyday life. As you can imagine, that just makes things worse and is way more harmful in the long run. So coming to this realization early on is not only what drove me to take a different path than my family did, but to make higher education a part of that journey. So I thought, going to community college was probably the first step towards getting into a career that seeks to combat this problem in some way. And that turned out to be probably the best decision I ever made. So To say that, it's been an uphill battle would be an understatement at this point. What's important is that it all started kind of with that vision, the interest or desire I had. And so for me, that's really what it took to get here. And it's what continues to push me even now as I face continued, challenges in the program or in my life. So at this phase in your training, I would encourage you to ask yourself what your driving force is and use that to ground you as you make decisions about graduate school and beyond. So for me, when I was applying to graduate school, it was really important that the program provided me with the training, the resources, the opportunities and the challenges to be, to allow me to be the very best version of a scientist that I could personally be. I'm happy to say that the University of Maryland and the PIN program was definitely the right choice in all aspects. For instance, it has, I think as Brian mentioned, it has an excellent selection of coursework, which brings everyone up to speed. My background was in psychology, so I had a lot to be brought up to speed about with regard to neuroscience, but it actually allowed me to, succeed regardless of the fact that I didn't really have a really strong neuro background. We had the opportunity to do multiple rotations, so you could find the best lab that suits you. The work being done here is very rigorous and impactful, so that's very important. And it's also very collaborative. So there's no limit to the experimental approaches you can learn or use. My project is probably a great example of that. I was interested in using electrophysiological approaches to investigate the synaptic mechanism of action for a rapid acting antidepressant that my mentor had discovered. So he encouraged me to actually do a rotation with one of his collaborators so I can learn and implement those approaches. And that work that I did, under their combined mentorship actually formed the basis of my current dissertation project. But something that definitely sets PIN apart, this was evident to me during the interview process. I interviewed at 8 different PhD programs. This is the one that actually had a focus in terms of providing financial support for students from underrepresented backgrounds, and that was the Meyerhoff program. So that just provides that extra level of community and support for students like us. For me, Meyerhoff has been a really important part of my graduate experience, not only because there are other students that I could identify with, but because it includes all students from the graduate program in life sciences, not just neuroscience. So it provides a great opportunity to discuss basically grad school experiences and research with the larger student body on a more regular basis. But probably what I found most valuable is that the Meyerhoff program serves as an advocate for its students, and especially if we were to encounter challenges in the lab or the classroom. So with the support from Meyerhoff and the strength of the PIN program overall that you guys heard, it really makes it a great program, and I could not recommend it more highly. Thanks.
So, are you going to tell us a little bit about your story?
Yes. So thank you, Lace, for sharing. Lace really hit all of the key points. Okay, so let me start off by introducing myself. My name is Saad Elzini. My pronouns are she, her, hers, and I am starting my second year here in the program. So I recently just chose my mentor and my lab, and I'm going down that path right now of my research, still starting my qualifying exams and stuff like that. I was born in Egypt and I was raised in Newport News, Virginia. And then I went to Virginia Tech for undergrad, where I actually just graduated in 2019. I did some undergrad research dealing with neurodevelopmental disorders and sex differences in that. So sort of like what Dr. Bale touched on earlier. And then I realized in my undergrad research that neurodevelopmental disorders and especially sex differences is something that I really wanted to focus and hone in on for my future research. However, financially speaking, I knew that grad school was something that was going to be very difficult for me. Having said that, I paid for my undergrad education and I worked to pay for my undergrad. So grad school just seemed very daunting. And then I looked into different types of grad school and I decided that a PhD program, although extremely competitive, would be sort of my best course of action for what I wanted to do. that being said, once I went through the interview process and sort of looked into all these different schools, something that really stuck out for me during my interview weekend at University of Maryland is the sense of community here that I didn't really find too prevalent in the different schools that I interviewed with. So sort of just this like really easy, laid back sense of community and communication between both students and faculty. You know, everyone's really just on like a first name basis and everyone's supporting each other. There's a lot of collaboration going on, and you really just felt like, you know, the students had a sense of hominess and a sense of real comfort to their research. So that being said, it was really competitive, which was something that scared me into getting into PhD programs. So once I did get that acceptance, you know, it was just that over the moon feeling. And then the imposter syndrome kicks in of, you know, am I good enough to be here? How did I get in? Oh my goodness. And, you know, all the self-doubt started creeping in. So something that I also appreciated about University of Maryland is that they really did a good job of presenting us with all the different resources that could be of help to me, both as a first generation college student, a person of color, and a person that, you know, like many of my other peers, were dealing with imposter syndrome, dealing with this, you know, first year in a PhD program. So that was always addressed in our orientation and supported by all the faculty. Georgia was, you know, one of the main people that just always had her door open for us. And, you know, we would go in, whether it be to vent, ask for help, ask her for advice, whatever it may be. So once I really like settled in and, you know, realized this is where I belong, this felt great. Meyerhoff program also reaffirmed that for me, of this is something that really made this place home for me, not only because of my passion for research, but also for the acceptance and sort of acknowledgement of myself as a person of color in the community. So the Meyerhoff program does a really good job of incorporating different talks of people from diverse backgrounds, helping you get in touch with people of diverse backgrounds in your field of interest, sort of making those connections that are really important. And I honestly didn't realize how important those connections were until, you know, you sort of look at the collaborations that are going on, the intricacies between, you know, different people that are working across the United States, across the state, just so many people that know each other and are helping each other with their research along the way. The reason I actually decided to choose my mentor and my PI right now, who is a white man, is because of something that he said while I was sort of speaking to him as a person that's interested in joining his lab. This was my second rotation, and he had said, you know, one of my main goals as your mentor is to help you get the connections that matter and help you get your foot in the door, acknowledging that I was both a woman and a woman of color, that it might be a little more difficult than anyone else that wants to get their foot in the door and wants to make all those important connections and succeed in academia. So that really just like, you know, was a moment for me of like, wow, this is someone that I want to be my mentor. This is someone that I know will be my cheerleader, be my person on the side, helping me get to all my goals, and then helping me push that ceiling even further and achieving things that I probably didn't even think was possible for myself. So honestly, really echoing what Lay said, this place has felt like a place that has helped me achieve my goals, my passions, as well as my dreams, and really just honing in on my capacity as a student, my capacity as a researcher, and improving myself day to day, both in science and in my personal life. So I really cannot sing any more praises of my time here, even though this is just my second year. And really, just don't get me wrong, a lot of your time in grad school may be filled with obstacles and struggles, but that's the whole point of, you know, realizing what you're capable of and pushing yourself past whatever it is that stands in front of you. So whether that be something that you grew up with or something that you're battling personally, a grad school program will really, you know, push you to your furthest limit, and then help you realize that you're capable of so much more than you thought you would be. I think that's all I have to say. However, I do want to make it, sorry about my dog. make a quick note that I am very available for any questions, whether it be personally to me as a student, as well as Georgia and the other staff. And if you guys have any questions for me, I'm more than happy to like help anyone at all that has any questions, comments, or concerns. I'm gonna mute myself now because my dog really wants attention.
Thanks a lot. That was great. Does anybody have any questions that I didn't already answer in the chat?
Any questions? This is a wonderful program and have wonderful students. I congratulate you all.
We absolutely do. Couldn't agree with you more.
Yes, yes, I'm very impressed. And I really am thankful that you asked me to be part of this seminar series. I really am. And I'm thankful also that I get to meet all of you and make sure that next year I'll invite you to talk to my students next year as well.
Excellent.
So any questions for the professors or the students? It seems that you guys, do you have internship opportunities for undergraduate students? Meaning summer internships, I guess, for research, or yes, they do have a summer research program, right? Did you guys mention that? I think that you did.
Yes, Jess, do you want to reiterate that?
So yeah, so we don't. So we have associations with other indoor programs. So I think we absolutely would have the ability to have a summer internship or a fellow come over this summer. In addition to that, I know that there are other opportunities through other pipeline programs that bring in undergraduates. And although I don't have that link at my fingertips right now, I will actually send it to Georgia so that she can send it around so that the students can potentially look at other avenues and opportunities to have funded research as undergraduates here at Maryland.
And I meant to ask you, so since you're so close to NIH, is there, they do have a prep program at NIH. Do you have collaborative, like students from the prep program from NIH could do research at UMB? Is it possible?
Brian, do you want to take this? I mean, I know that anything is possible, right? In terms of collaboration. But I am not aware of any sort of NIH funded students coming to University of Maryland. They typically, in my experience, I've seen them stay at the NIH institutes. However, that doesn't necessarily mean that we don't have the collaborations with them coming to our labs to do some work. But I don't know, Brian, if you know anything else about that.
I can mention something really quick. So I know NIDA at least will always invite us to take on, they have a summer program, and students at our university have been invited to participate in that. Actually, I have a form I have to go fill out online. So there are, I guess, that through their summer programs, we can host students from at least from NIDA, which is the National Institute of Drug Abuse. I don't really know about other areas, whether other NIH institutes do that as well.
Yeah, I'll just finish up by saying that NIDA, National Institute of Drug Abuse, is in Baltimore. It's about 2 miles away from us. And that, like Jess said, anything is possible. It's one of the great parts about being in Baltimore is there's so much neuroscience across Hopkins. NIDA, National Institute of Aging, is in Baltimore as well. And the main campus of NIH is like 30, 40 minutes away. So there's just a critical mass of research going on, and there's a lot of collaborative efforts between these institutions as well.
And I think Brian just raised another good point, is that the flexibility. One of the things that I really have to commend Maryland and our program in particular on is being flexible. So we're not very rigid in terms of requirements. And if you can sort of think it, we can find a way of doing it. I think we work very hard at tailoring the education of our students.
Well, I also want to add that our Neuro-ID program, our Neuro-ID students, if they find a lab at any institution, we have funds to pay the summer program. So if one of our students, which seem to be very interested in your program, are really looking forward to make connections and talk to Georgia or Brian, I can say that if you're part of our program, we'll pay your way. So it's just a matter of making the the connection and the lab and all the network that is needed for that. So rest assured that that could also be managed. So once again, I feel that we are all in the same page. That we are in the profession of advocating for our students for their development and also for achieving their goals. And so I really appreciate all your time and being here with us here. And of course, we'll stay in touch. I know we will. So you will have students contacting you. Do we have your emails and everything? And since now it's 6:30, I think we can say thank you so much for being here. Tracy, thank you for a wonderful talk. Brian, Georgia, for keeping us, through the email, communication through these months. And of course, it was a pleasure to meet Jessica and Mary Kay. It's a pleasure. And of course, Lace and Sod. It was wonderful. Thank you so much. And I hope to see you back.
Dr. Maldonado.
Yes, Hector.
Briefly, on behalf of all the Neuro-ID students, and while the rest of the faculty of the University of Maryland is still here, we wanted to congratulate you for your recent award, the Bernice Grafstein Award, because of your outstanding mentorship. And just generally thank you for your commitment to promoting a more inclusive form of science. Y I'm going to say this part in Spanish, por ser, ¿verdad? Por un orgullo boricua. So just thank you for everything.
Thank you, Hector. Gracias.
Thank you.
Yay! Congrats!
Gracias, Luisa. Thank you so much. You see, I always say that the students take all the credit. We're just, facilitators in this process. So thank you so much, everyone. I really appreciate those comments. And again, thank you all for being here. And I look forward to see you next week because this continues to December. Every Wednesday at 5. Even though I forget English and Spanish sometime and I start mixing them up, it doesn't matter, but we're doing this. I'm committed. So you take care of yourselves and have a wonderful evening. And I'll see you guys later.
Thank you, Clara. Thank you. Hasta luego.
Bye.
Bye.